First.
Now please, good evening बच्चों, good evening. Am I audible now? Okay. So good evening everyone, and please tell me what was the last thing we have done so that I can continue from there. The last thing that we have done. Please write down in the chat box. At least 1 or 2% can write so that I can gauge.
Sir, tell us the syllabus is about.
हाँ बच्चों, so digestive glands are completed. So intestinal gland completed, and I gave you that table to make. Did I give you? Did I give you the table to draw? Okay, so did I ask? I didn't ask you for that. क्या हो रहा है यहाँ? For example, I ask you to make this type of table of all the digestive juices. We know that we have 5 type of digestive juices, starting from saliva, starting 1, 2, 3, 4, 3, 4. Starting from saliva, then gastric juice, then bile, pancreatic juice, and succus entericus. Now just see in your notebook, we have done this one. Raghunandan is saying no, Oppo is saying covered. At least 2-3 more people proceed is saying done. Because I can remember this thing was completed. I told you done. Okay, Joshita Karthik saying done. हाँ बच्चों, check it. Now coming now, good. So, beta, very patiently, if you tell me the truth, so that we can proceed. Siddhi, Karthik, we have made this one or not? If we have made this one, we can, Priya, we have done this one. Very good. Now we will start from mechanism of digestion. Means digestive glands are over now. Now we have to do mechanism of digestion. Let us begin. It's a very interesting thing and a very easy thing also. We have to do mechanism of digestion. After that, we will do the absorption. Absorption only we will do assimilation, and also egestion. Defecation also will be done in this section only. And then we will do the calorific values, then we will be doing the disorders. In this order we'll be doing, okay? After absorption, we'll be doing the calorific values and disorders. And in disorders only, we'll be doing the protein energy malnutrition. And the class will get over by discussing about the PEM. So this will be our, today's plan, starting with the mechanism of digestion. Shall we proceed? Very good. Now, whatever you have eaten in the afternoon, in your lunch time, mostly we take pappu, which is mainly rich in proteins. We take rice annam, which is mainly rich in starch. Some chutney, which is a combined thing, and some curry. Curry has all minerals. It has got proteins also. Some of them have proteins and carbohydrates, again, in those curries, plus oil. We use oil for cooking. So oil means triglycerides. We are taking fats. So nearly all the components, however, in Indian diet, we are more, dependent on starch. As we all know, we are more dependent on starch. Where are the places where digestion takes place? So the sites of digestion, first of all, digestion occurs only in 3 places in the body. One is in the buccal cavity. Second time it occurs in the gastric or the stomach. In the stomach it happens. And then the last one in the small intestine. Till here. And in these 3 sites, digestion takes place. Are you guys with me? Buccal cavity, stomach, and small intestine. In the buccal cavity, it occurs with the help of saliva. In the stomach, it is done with the help of gastric juice. And in the small intestine, it is done in 2 steps. Step number 1 and step number 2. In the first step in the small intestine, what will act on the food is pancreatic juice. Along with bile. And next, succus entericus. So this completes the digestion in the small intestine. But the small intestine is the main site. It is a main site for digestion. And we will see that it is also the main site for absorption. Maximum absorption occurs in the small intestine. Are you guys with me so far? Very good. The pH in the buccal cavity is maintained at 6.8. In the stomach, it is about 1.8. And here it is maintained at nearly 7.8. So these are the pH at which the digestion takes place. Almost neutral, acidic, and it is slightly alkaline. Enzymes also, in the stomach, in the saliva, we have got salivary amylase. Now this I am giving as a homework. Salivary amylase in the gastric juice. You have to tell me 3 enzymes. The third one I am writing the name is gastric lipase, which is not very active, so I am not discussing it right now. But yes, we should know the presence. Second one is especially present in infant and children. infant and children. So therefore I put a conditions apply here, and the first one is present in the POTA, P for POTA and P for that enzyme. So 1, 2, 3, the third one I am saying it is not very effective in the human beings. The pancreatic juice, we have got 6 enzymes in the pancreatic juice, and then succus entericus also 8 enzymes in the succus entericus. The previous chart will help us to remember the enzymes of pancreatic juice and the enzymes of succus entericus. Are you guys with me so far? Focus all your attention on the buccal cavity where the digestion takes place. You should also note down that digestion takes place by both mechanical and chemical. Chemical digestion takes place with the help of enzymes. Enzymes cause hydrolysis of the food products. Hydrolysis means breaking with the help of water. Breaking with the help of water. So I am now focusing all your attention on the first one, buccal cavity. Please understand. Now in the buccal cavity already you know that saliva is acting, and saliva has got salivary amylase. So I'll write that title, digestion in, you can also write down like this, digestion in buccal cavity. Digestion in buccal cavity, and here the food component which is digested, carbohydrates. Carbohydrates, we have got starch. Starch is digested with the help of salivary amylase. You know, bachcho, we have a salivary amylase here. Salivary amylase at a pH of, you already know, the pH. Starch is broken down into maltose, maltose and isomaltose. They are almost same, isomaltose. In maltose, the bond is different, so these bonds are different, you know. This is alpha 1,4 glycosidic bond. This is alpha 1,6 glycosidic bond. Otherwise, both are the disaccharides. Beta, maltose and isomaltose are disaccharides. So carbohydrate digestion takes place in the buccal cavity, but again a conditions apply here. Okay, and what is this conditions apply here means that it is up to it is up to 30%, not more than that. The digestion in the buccal cavity is up to 30%. You have seen in the Shara time that there are many sale coming on the store, on various store. It is written up to 50%, up to 30%, but that is up to conditions apply, and the condition apply is it is up to that, not necessarily 30. It can be 10 also, but maximum it is 30. Here also I am saying the same condition, up to 30% starch can be digested in the buccal cavity. Conditions are starch should be properly cooked, and yeah, we can chew it properly. Then these are the two conditions. We can digest up to that too. If it is properly cooked and we chew it properly, up to 30%. Well, the next step of digestion occurs in the stomach. So buccal cavity, if someone asks you buccal cavity, other things, proteins, digestion of proteins, digestion of fats, and digestion of nucleic acids. Digestion of nucleic acids, absent. No digestion of protein, fats, and nucleic acids. Next one. This is digestion in stomach. Very easy. Digestion in stomach. Acidic pH, enzymes are pepsinogen and pepsin, which is the active form of pepsinogen. You should also don't forget that pepsinogen, how pepsinogen is activated into pepsin. Who will tell me what will activate pepsinogen to pepsin? Yes, please. Yes, very good. It is hydrochloric acid. Very good. Don't write enterokinase for heaven's sake. That was for trypsinogen, bacha. Suhas, it is for trypsinogen. Okay, beta. Pepsinogen, so hydrochloric acid. Moreover, it itself is also autocatalytic. So pepsinogen is activated by HCl and also by pepsin. Some active pepsin is there. It can also activate. Same is the case. Please don't forget the prorenin can also be activated by the same pepsin into renin. So these are the active form. Now, you know that proteins are digested by them. So proteins, the proteins in the diet digested with the help of pepsin, into peptones, peptones plus proteoses, peptones and proteoses, peptones and proteoses. What are these? These are smaller peptides. These are smaller peptides. Okay. What are these? These are smaller peptides. A protein is a very long chain of amino acid, and then it is broken into smaller chains. Okay. So these smaller chains are given the name as peptones and proteoses. Pepsin digests proteins into peptones and proteoses. Remember, bachcho, pepsin is an endopeptidase. It can digest up to peptones and proteoses. Don't expect amino acid to come directly. You know that the karam puri is basically made from dried chilies, okay? So you have got dried chili and you beat them to make karam puri. And you have started beating them. Don't expect that a couple of beating you start and directly you get the puri. No. In the first, it will broken into 2 pieces, then more pieces, and then smaller and smaller pieces. Same happening here. You can see directly it won't yield you amino acid, it yield you peptones and proteoses. Please be careful, this is proteoses. The word proteose is a peptide. However, the word protease, if the word protease is given, as you know that ase is an enzyme, this means a protein digesting enzyme. Protein digesting enzyme. So also be careful about the spellings. Also be careful about the spellings. So proteose is a peptide. However, protease means a protein digesting enzyme. As we know, A's are enzymes. But in the stomach, there is one more enzyme, is renin. And what the renin does, renin converts milk into curd. Once again, renin converts milk into curd, perugu, curd. In the milk, there is a milk protein called casein. The white of the curd is actually calcium para-caseinate. The white of the curd is calcium para-caseinate. I should call it calcium. The right pronunciation is calcium, magnesium, potassium. We used to pronounce it wrong, potassium. I found that it is not potassium, it is potassium. Anyways, and this is done with the help of renin. Renin, and here calcium ions are also required. And milk has plenty of calcium ions. Where is this calcium para-caseinate? How do you notice, how do you find that? You take curd and you hang it in cloth. And then what you get, the water will come out, and what that semi-solid stuff that you get, that is calcium para-caseinate. So you make curd outside, okay? We know in kitchen we make curd, but in the body also curd is made, and this step is called curdling. Curdling of milk. This step is called curdling of milk. But so if the question comes, which enzyme help in the curdling of milk? Answer is renin. So simple. Now this calcium para-caseinate is acted upon by pepsin. And what you get are peptones. Peptones plus proteoses, okay? Now you get these peptides. Unless it is formed, it cannot be digested. So we can say that renin help in the digestion of milk protein. Be careful, it is a milk protein. Casein is a milk protein, not milk sugar. It is a milk protein. Make sense? Understood? Everyone got it? Fine. Proceed. In the stomach, nothing else. So carbs are not digested in the stomach. Then fats are also not digested. Though we have a lipase, but emulsification is not there. So, and that too, gastric lipase is also in very small quantity, very small quantity, very small quantity, and that too in children, in infants, okay? So we can say infants and children, young 0 to 5 years of age. So don't expect any fat digestion here, and nucleic acids also not digested. So these three are not digested in the stomach. In the stomach, proteins are digested. So two places, one is in the buccal cavity, carbs. In the stomach, proteins are digested. Now let's come to the third place, which is small intestine. And first time, a complete digestive juice will be present in the small intestine. Pancreatic juice is having maximum diversity. In terms of the enzyme, highest diversity can be noted in pancreatic enzyme. Even in case of, because you know you will not find the amylase in Succus entericus, even the lipase in Succus entericus is questionable. So if a question comes, name the digestive juice with maximum diversity of enzyme, pancreatic juice. Shall I write here? Better digestion in small intestine, small digestion in small intestine, please note down. The first step, number one, step one is the action of pancreatic juice, action of pancreatic juice and bile. Both are acting, action of pancreatic juice and bile. Let us see. In the pancreatic juice, all enzymes are there. You have got trypsin, so trypsinogen. Who will tell me the answer? Suhas, will you like to try? What will activate trypsinogen to trypsin? Come on, trypsinogen to trypsin. Anyone can tell me how this activation takes place? Yes, you are right. This answer is enterokinase. Enterokinase. Perfect. Now, in which digestive juice you find enterokinase? Who will tell me the answer? Where will you find enterokinase? Intestinal juice, very good answer. Intestinal juice. It is an enzyme of intestinal juice. Very, very good. And yes, you are right. It is also autocatalytic. So, but in the examination, the teacher is actually asking enterokinase. So yes, if an option is enterokinase plus trypsin, the best answer. Otherwise, enterokinase is the answer. But similarly, we have got pancreatic lipase. Can you please tell me who activates activated lipase? Now, let us see who will answer this question. How lipase is activated? Although it is not a zymogen, it is not like liposa. No, this is not lipids digesting, but it is lazy. In presence of dash, lipase become more active. What is that? Who will tell me the answer? Yes, Suhas Gowda has given the correct answer. And here the answer is bile salts. Very, very good. Bile salts activate the lipase. If I make the paper, I will give this question. Bile salts activate the lipase. Chymotrypsinogen is activated by trypsin only, and procarboxypeptidase is also activated by trypsin. It is an autocatalytic enzyme. But in step number one, we have to see the action of pancreatic juice. Let us take food components one by one, starting with the carbs. Carbohydrate, we take starch. Do we take glycogen also? Yes. When we take glycogen, when we eat non-vegetarian food, glycogen. These are digested with the help of amylase. With the help of amylase, pH of alkaline pH of 7.8. And what we get? We get same maltose, plus isomaltose. Disaccharides, maltose plus isomaltose. Carbo is over. Let us come to the proteins digestion with the help of pancreatic enzymes. Let us write proteins by enzymes like trypsin, like chymotrypsin, trypsin and chymotrypsin. It will make peptones, peptones plus peptides. Now what? Now this will be acted upon by another enzyme. This will be acted upon by another enzyme, and that is very important to note down here. This is activated by another enzyme here, and this will give you dipeptides. Dipeptides. Okay. Anyone can tell me the name of the enzyme? It is easy now. It is easy because two names I have already given you, and the third one is left now. Please tell me. Yes, it is carboxypeptidase. Yes, you are right. It is carboxypeptidase. We get dipeptides. So in the small intestine, proteins are digested to dipeptides. Carbs are digested to maltose and isomaltose. We are seeing the action of pancreatic juice and bile. Now, next one. Let us talk about the digestion of fats. Now, dietary fats are triglycerides. We eat triglycerides. Triglycerides are present in oils, butter, ghee, neigh, all contains triglyceride. Who can define triglyceride? I know it is difficult to type. You can just, in short, the moment you write something, I will understand that you understand or not. What are triglycerides? Very good. Suhas is correct. Glycerol plus 3 fatty acid. Triglycerides are 1 glycerol. अरे क्या है यार, ये क्या हो गया है मेरे फोन में यार? Any time it starts saying something. Glyco.
Section 4 boy came.
Section 4 boy came. Okay. I'm coming outside. Coming. Okay. Triglycerides are esters of 3 molecules of fatty acid and 1 molecule of glycerol. Esters of 3 molecules of fatty acid and 1 molecule of glycerol. Triglycerides. What are these? These are esters of 3 molecule of fatty acid and 1 molecule of glycerol. 3 molecule of fatty acid and 1 molecule of glycerol. And whenever an acid and alcohol join, the bond formed is ester bond. So these are esters. And our dietary fats, these are the dietary fats. We eat them. Now, how to see their digestion? But first, let us understand the problem with the lipase. Lipase are the enzymes to digest them. And we know that lipase can be activated by bile salt. Okay. To understand the problem of lipids, let us take lipids in a beaker, or you can take them in a test tube. I'll give you one example, which you will also see in your first year of medical college. In the biochemistry laboratory, you will find this experiment. You will be given test tube and some oil will be given. You can take some water here and oil. You find that the oil is floating. Obviously, the oil is floating on the surface. Okay. Now, what will you do here? You will add some bile salts. You will sprinkle some bile salt. Same like uppu, little greenish in color, we get bile salt. We take it and just sprinkle, and then the magic begins. In that magic, this all come down. This will start melting down. All the oil will melt down, and after some time, what we get is an emulsion, a homogeneous mixture. We get an emulsion of water and fats. What has happened here? The big fat has broken into small, tiny droplets. This is an emulsion. This step is called emulsification. The same step happens in our gut also. What is the advantage with this? Advantage with this is the surface area has increased too much. By emulsification surface area, you can imagine a big drop when it is broken into tiny droplets. Suppose if it is broken into small drops, and you take the surface area of all the three smaller one or all the four smaller one and take cumulative surface area, the cumulative surface area is much higher than the surface area of this drop. Maybe this is obtained from this. This is the product of breakdown of this only. But surface area increases. And bachcho, you should know that the lipases, the enzymes require surface area to act. Enzymes, they have to bind to the substrate. Unless they get an area, they cannot bind to the substrate. And unless they bind to the substrate, you will not find their action. So this step is very important. This same thing, what is happening in the test tube, happens in our intestine also. Just imagine our small intestine, this is having watery medium. It is having watery medium, all watery. Aqueous medium, all is aqueous medium. Aqueous medium, you understand? All watery medium. So the problem, you understand, same problem is going to happen, but thanks to bile salt will convert this into an emulsion, tiny droplets, so that lipase can easily digest them. You understand now? You got it? Why? So fats, first step in digestion is emulsification, that is done by bile salts. Bile salts turn them into an emulsion. Fats plus water turn into an emulsion. Emulsion surface area has increased. And now in the emulsion, the lipases will act. Don't forget that fats contain triglycerides. In the first step, you will find that it is broken into a diglyceride and one fatty acid. Again, it is broken by lipase. The same lipase will act again, and diglyceride is broken down into monoglyceride, and one fatty acid is released. So you can understand that two fatty acids can come out by converting triglyceride into monoglyceride. Okay, so because triglyceride is an ester of 3 molecules of fatty acid, this is what happens in the, this is a pancreatic lipase. Let me write down, this is pancreatic lipase, step by step. Second step, and what we get is monoglyceride plus fatty acid. So triglycerides become monoglyceride and fatty acid, and thanks to the bile salt, the main digestion occurs not by the bile salt, but by the lipases. The enzyme is lipase and not the bile salt. And remember, these are bile salt, not bile pigment. What is the role of bile pigment? No role. Bile pigments are waste products. They're excretory waste, bachcho. Yes, you are right, Joshita. Okay, but so now triglycerides, let me quickly revise your uh, pancreatic action of pancreatic juice and bile. Trypsinogen activated by enterokinase into trypsin. It is also autocatalytic. Lipase activated by bile salts. Okay, now let us start the process. Carbohydrate, starch or glycogen acted upon by pancreatic amylase broken into maltose and isomaltose. Proteins by trypsin or chymotrypsin into peptones and peptides, and then by carboxypeptidase into dipeptides. So ultimately, we get dimer of amino acids. This is the problem with the fats, and what is an emulsion? The fat is broken down into tiny, tiny droplets. As a result, the fat surface area has increased. The same problem that happens in the test tube also happens in our small intestine, thanks to the bile salt which are converting the fat into an emulsion. And then in the emulsion, the lipase become active. Lipase act on the triglyceride and break two times into two fatty acid and monoglyceride. I think this is simple. And lastly, digestion of nucleic acids. What are nucleic acids? Nucleic acids. First of all, bachcho, remember that nucleic acids are DNA and RNA. Na? They are nucleic acids are DNA and RNA. Okay, understood? Good. Our digestive system can also digest them. This is efficiency. We can even take nutrients from DNA and RNA, okay? Because food contains the cell of plants, animals, and in the cell nucleus is there. In the nucleus, plenty of DNA and RNA is present, so we can digest. We have nucleases, enzymes called nucleases in the pancreatic juice, which will digest them into nucleotides. Nucleotides. So, bachcho, this is what happens in step number one. Step number one is over. Step one is over. And now starts step number two. Now starts step number two. In step number two, action of succus entericus. Action of succus entericus. Succus entericus. Bachcho, this is the last step of digestion. Let us take carbohydrates, carbs. In the carbs, now we have plenty of disaccharides. For example, you have got maltose, you have got isomaltose. How they are formed? By digestion of starch and glycogen. We have got sucrose. How sucrose formed? Sucrose is added into the food. It is a panchadara. It is the table sugar. We have got lactose. From where lactose come? Lactose is a milk sugar. It comes in milk. These are digested by enzymes like maltase, like isomaltase, maltase, isomaltase, sucrase, lactase. We get two molecules of glucose. Here also two molecules of glucose. Now sucrose, we get one glucose plus one dash, and lactose, we get one glucose plus one dash. Can you please tell me the sucrose dash? What should, yes, it is fructose. Very good. This is fructose. Good. And can you tell me the dash of lactose? Yes, you are right. It is galactose. Galactose. Now, which one is a monosaccharide? All are monosaccharides. All are simple sugars, and they are ready for absorption. Absorption. They are ready for absorption. All are monosaccharides, and now they are going for absorption. Carbohydrate digestion is completed in the small intestine by step number two, and here action of succus entericus enzymes are occurring. Now, let me take you to the proteins. Proteins, what will happen here? Proteins by dipeptidase. Dipeptidase. And what you get are two amino acids. to amino acids. This is also ready for absorption. This is ready for absorption. We can absorb amino acids. Okay. Now what? After that, after proteins, now we will take fats. In the fats, we have got monoglycerides. बच्चों monoglycerides. Let me take you proteins here. Proteins. In the proteins, we have got dipeptides. Dipeptides. You know dipeptides, how they are formed? Action of carboxypeptidase on peptones. We get dipeptides. Dipeptides by dipeptidase. This is the protein digestion. Fats, monoglyceride. Monoglyceride acted upon by lipase. This is intestinal lipase, bachcho. And what you get is glycerol. Glycerol and one more thing, dash. Glycerol plus dash. What is this dash? Glycerol plus dash. Fatty acid. Good. Glycerol and fatty acid. Very good. But so after that, after fats, nucleic acids. In nucleic acids, we have got nucleotides. Now, nucleotides by nucleotidase, we get nucleosides. Now what? Nucleosidase. And what we get? We get pentose sugars plus nitrogen bases, purines and pyrimidines. And now they are ready for absorption. They are also ready for absorption. Pentose sugar and nitrogen bases are ready for absorption. In monoglycerides, when intestinal lipase digests them, what we get? Glycerol plus the last thing. Yes, you are right, it is fatty acid. Very good, fatty acid here, fatty acid. And now this is also ready for absorption. So now all the food products are ready for absorption. Carbohydrates, you have got glucose, fructose, galactose. In case of proteins, amino acids are there. In case of fats, we have fatty acids and glycerol. In case of nucleic acids, means DNA, RNA, we have got nitrogen bases, purines, pyrimidines, plus a monosaccharide, pentose sugar, ribose, deoxyribose. These are pentose sugar. Pentose matlab 5 carbon. Glucose is 6 carbon. Okay, bachcho? This is the mechanism of digestion. After mechanism of digestion, you have to learn the mechanism of absorption. Okay, are you guys with me so far? Are you guys with me? Okay, very quickly, these 12 pages, I can share with you all these. In the beginning, I was just asking you what we have done. Then our target today, we are here. We have completed up to here. Okay, then mechanism of digestion, the sites of digestion, these 3 sites. After that, in the buccal cavity, mainly carbohydrates are digested into maltose and isomaltose. In the stomach, mainly proteins are digested. And you can see the pepsinogen active by HCl into pepsin, proteins into peptones and proteoses. These are smaller peptides. The milk protein is curdled. There is curdling. Curdling means making milk. Converting the milk into curd. Converting milk into curd is curdling. But I'll write one note here, very, very important note. Now, the note is, in adults, comma, curdling of milk occur by the help of which enzyme? Which enzyme help in the curdling of milk in adults? Come on. Adults don't have renin, bachcho. Adults don't have renin. So when I am taking milk, you are taking milk. What is in the digestive system, or which enzyme help in the digestion or curdling? And the answer is chymotrypsin. Please learn. It is chymotrypsin. Chymotrypsin. Learn the answer, chymotrypsin. Adults, the same thing is done with the help of chymotrypsin. Okay, now carbohydrates, gastric lipase, I said no action as such. In the stomach, carb, fats, and nucleic acids are not digested. Now, small intestine, step number 1, action of pancreatic juice and bile. And bile, mainly bile salts in the bile are required for digestion and absorption of fats. Beta, trypsinogen active by enterokinase into active trypsin, and this is a very, very important question. Lipase also activated by bile salt. Let us see carbohydrate, the same maltose and isomaltose, proteins into peptones and proteoses. Again, there is an exopeptidase by the name carboxypeptidase. It is a different type of peptidase. Both are almost the same type of peptidase. It's a different type. It can convert it into dipeptides. Then fats. In the fats, I told you, first of all, emulsification takes place with the help of bile salt. Now, in the emulsion, lipase act, and we get monoglycerides and fatty acids. In nucleic acids, we get nucleotides because many nucleotides are joined by phosphodiesterase bonds, right? And these bonds are digested by nucleases. We can have, for DNA, we have a DNAase. We can call it as DNAase. For RNA, we have got RNAase. Understood? After that, step number 2, action of succus entericus. Now, in succus entericus, we know the enzymes. I told you the trick to remember Dan Mills, okay, this is again I'm reminding you. So see, carbohydrates, maltase, isomaltase, sucrase, lactase are digesting the sugars, disaccharides into monosaccharides. After that, dipeptides, you know that pancreatic enzymes have made up to dipeptides. The dipeptidase is present in succus entericus, and we get to amino acids. Fats, we have monoglycerides left in the pancreatic digestion, and now the intestinal lipase will convert it into glycerol and fatty acid, and nucleotides are the product of digestion by the pancreatic nuclease, and now the nucleotidase and nucleosidase will digest them up to pentose sugar and nitrogen bases. So this is the culmination or completion of digestion. But so after digestion, what happens in holozoic nutrition is absorption. After digestion, what happens is absorption. And this is also important thing to learn here. Have you understood this much? Any issue here up to here? Any issue you can tell me? The YouTube children cannot say, but other children can say. Any issue you have, you can say. Tomorrow exam is there.
The intermediate, they have to learn it.
Tomorrow exam.
They have to the exams are long term.
2 exam. Long term. Are you having 2 exams this week? Are you having 2 exams this week? One exam only. One only, बच्चा one बोल रहा है, check कर लो जरा. I also remember one only. पेपर ही वन बनाए तो फिर 2 कैसे हो जाएंगे? बच्चों, now absorption. हाँ, इनको लाइव कर दो. If they give which enzyme digest milk protein, What is the answer? Renin. Anything about the milk, answer renin only. Very, very basic. Renin. Any other doubt, bachcho? It's a good question. Windows 7 has asked. Okay. Okay. Now, what are the places where absorption takes place? Absorption takes place everywhere, starting from the buccal cavity, stomach, and small intestine and large intestine. In every place, absorption takes place. So the sites of absorption are, I'll discuss all of them, starting with the buccal cavity. Buccal cavity, after that stomach, small intestine, and large intestine. Small intestine and large intestine. Buccal cavity, stomach, small intestine, and large intestine. Small intestine is the main site for absorption also. This is the main site for absorption. Now let me tell you what are the buccal cavity. Buccal cavity, see, remember D here and D here. And what this D stands for? D stands for drugs. So drugs are absorbed. You know that some drugs are kept underneath the tongue, sublingual, underneath the tongue. And some drugs are enteric coated, means that when they reach the large intestine, there they are absorbed. They are made into that way. However, in the summary, you will find that some drugs can also be absorbed in the acidic medium of stomach. So, normally we will not answer that way. We will answer only D and D, front and back. But if a question is asked in that way, what are the sites, and that includes stomach also, then we will say all of these. Then the answer is all of these, the best answer. That in buccal cavity also, yes, drugs can be absorbed below the tongue. In the stomach, acidic medium, few drugs are absorbed. In the large intestine, yes, drugs are absorbed. But right now, I'm saying D there and D here. In the stomach, just remember, wash, W-A-S-H. Now, W stands for water, A for alcohol, and S for sugars. These are the simple sugars, monosaccharides, alcohol, sugar in the stomach. In the large intestine, DMW. You have heard about BMW? Our is DMW. DMW, drugs, M for minerals, and W for water. Among the three, water is the main thing that is absorbed in the large intestine. Among the three, water is the main thing that is absorbed in the large intestine. But in terms of the quantity, mainly it is absorbed, or maximum. If a question comes like maximum water, if you take water, then in which place maximum absorption takes place? In a day, small intestine, bacha, this is the main site for, it is 6 meters long, 6 meters long, and then it has got villi and microvilli. Villi and microvilli. You can understand how much big surface area. It is the best suitable for absorption. And rest all, everything that can be absorbed is absorbed in the small intestine. Everything that can be absorbed is absorbed in the small intestine. Now, let me take you to the mechanism. Okay, this is one table given in the textbook. You can also see that. However, you can learn easy. Okay, DD, DMW, WASH. Okay, so 1 or 2 questions can be answered accordingly. Mechanism of absorption. Everyone, including the offline students also, just check your notebooks. If not, then you can write down. we have got two mechanisms. Basically, we have got active transports, and we have got passive transports, passive transport. Okay. Now, why we are talking about membrane transport? We are discussing absorption, then why we are discussing about membrane transport? Let us understand. See, when we take something in the diet, it is absorbed, and it is absorbed in the intestines or wherever in the mucosa, right? And mucosa is made up of epithelium. Let us say in the small intestine. In the small intestine, we have got the epithelial cell having microvilli. I'm just taking an example. So, if something has to be absorbed, let us say if fats have to be absorbed, or let us say sugar has to be absorbed. So, sugar or glucose is coming here, and then it can go to the blood. After that, it can go to the blood capillary. So you can understand that how many times it is crossing the plasma membrane. Sugar come here, after that sugar, this glucose comes here, and then it enters into here. So without understanding the membrane transport, we have to include membrane transport in absorption. Okay. Now, active transport, you know that it is against the concentration gradient. It is uphill until it is against the concentration gradient. And passive transport is along the concentration gradient. Active transport requires ATPs, and passive transport is independent. It is independent of ATPs. Bachon. Now let me take you some names of passive transport. For example, simple diffusion. For example, simple diffusion. For example, number one, number two, osmosis, water osmosis. Number three, facilitated transport. Why we are saying it is a simple diffusion, but it is called facilitated because it requires a carrier protein. Note down this: active, passive, active against the gradient, passive along the gradient. In the passive, some names are there like simple diffusion, osmosis, water in the favor of the concentration gradient, the osmotic gradient, and then we have facilitated transport. This requires a carrier protein. Right now, some of the passive transports are actually indirectly active. Some passive transports are indirectly active, or we can say, for example, this facilitated transport is secondarily active. It is secondarily active transport. Means what? Means if you stop the supply of ATP, it will also stop. Right? But when you are just focusing on that mechanism, it is facilitated. It is not showing any change in the protein conformation. It is not showing that, right? It is that particular name, facilitated, is a passive transport, but ATPs are required. To draw the ions inside, you have to create a concentration gradient. Let us understand this only. And this is the only problem that we have in understanding the absorption. I think I gave you some of the passive are actually secondarily active. Okay, let us go to the next page. Let me take you to the absorption of glucose and amino acids. Let me show the epithelial cell. We have got some carrier proteins for absorption of glucose and carrier proteins for absorption of amino acids. This is a blood capillary. It's a blood capillary. Let us understand the mechanism. Now, we have a carrier protein which is favoring co-transport. Means what? It can transport two sodium ions, but along with that, it can transport the glucose molecule. You understand? It is a co-transport. It is a symport. Co-transport or symport together. This carrier protein facilitates both. Unless this comes, this will also not come. If sodium is not coming inside, glucose can also not be absorbed. So it is a co-transport. Sodium is a co-transporter, and this is called facilitated transport. The name here we will use is facilitated diffusion or facilitated transport. It is an exchange, it is a symport, it is a facilitated transport. But so please understand that to do this, we have to create a gradient, a concentration gradient, so that, you have got more sodium in the lumen as compared to the cell. Sodium should be high in the lumen and lower in the cell. But you can see that when you are taking sodium inside, sodium ions are accumulated inside and the gradient can be lost. So how this gradient can be maintained so that glucose can come inside? The gradient can be maintained by an active transport mechanism happening here. This is sodium potassium exchange pump. It can pump in potassium and pump out sodium. This is the same Nokia. Okay, the same Nokia. Sodium को out किया, potassium को in किया, Nokia. Sodium out, potassium in. Sodium out of the cell, potassium inside the cell. Beta, this is an active transport. This requires ATP. This is ATP dependent, and this is creating a concentration gradient inside the cell. And because of the concentration gradient, sodium ions are attracted inside, and when sodium ions are attracted inside, along with them glucose molecule is also coming. If sodium do not come inside, glucose also cannot come inside. You understand? So facilitated transport actually indirectly depends upon this active transport. We can call it secondary active. Secondary active. This is a secondary active mechanism. But so this is possible, this is occurring for glucose and amino acids. I hope. And now coming, this is the mechanism I have explained you. And now finally, what you have to write in the examination. Okay, done. Once again, active mechanism and passive mechanism. 3 names to remember here: simple diffusion, osmosis, and facilitated transport. Coming here, I have explained how come this particular thing is facilitated transport. Then how it is dependent on ATP. It is dependent on ATP secondarily. If you stop the ATP supply, the gradient will be lost and glucose movement also stop. It is a co-transport. We can call it a symport. It is a symport. It is a co-transport. But now, like substances, number 1, glucose. Glucose and other monosaccharides. Glucose and other monosaccharides, how it can be absorbed? It can be absorbed by 2 mechanisms. Number 1, simple diffusion. Simple diffusion. This is along concentration gradient, and then facilitated diffusion, facilitated transport, and this is actually secondary active. This is actually secondary active transport. So we cannot ignore that this is dependent on ATP or active transport. The glucose absorption in the intestine occur, number 1, with the help of number 1, simple diffusion, number 2, facilitated transport, number 3, active transport, and number 4, All of the above. All of the above are responsible for absorption of glucose and other monosaccharides, and this is same applicable for many amino acids. Amino acids. But please note down sodium ions. Sodium ions are absorbed by active transport. Active transport. But chloride ion, passive transport or simple diffusion. It is by simple diffusion. Simple diffusion. Chloride by simple diffusion. Water. Water by osmosis. Simple diffusion. Simple diffusion. What else? So I have taken, and lastly, fats. Fats again by simple diffusion. Fats by simple diffusion. Simple diffusion. So active transport is sugars, amino acids. Sugars means monosaccharides, active. Sodium ion is only active. Chloride is passive. Water passive. Fats passive. These three are passive. These three are passive. Glucose is both passive as well as secondary active. It is both passive and secondary active. But to note down this one, this can give you answer like how water is absorbed, how fats are absorbed by passive diffusion, simple diffusion. And lastly, very, very important thing I'm going to discuss, very, very important, the absorption of fats, mechanism of absorption of fats. Now, what fats we eat? We eat triglycerides, and when they are digested, after digestion, what we get? We get fatty acids, okay? We get fatty acids, and what we get? We get glycerol, fatty acid and glycerol. Glycerol, fatty acid and glycerol. And they can be absorbed passively. But the problem is, they can be absorbed through villi. And the problem is how they go to the villi. Understand? If you take a test tube or a beaker and take water into it, and you add fatty acid into it, what do you find? That fatty acid starts floating on the surface. Fatty acid will start floating on the surface. They will form a single layer, monolayer, like this. They will form a monolayer, like this. They will start floating. They have got a hydrophilic head and they have a hydrophobic tail. I am showing the tail by pink color. It is a hydrophobic tail. I am showing the head by blue color. It is hydrophilic and the tail is hydrophobic. It has got a zigzag tail which is hydrophobic, and it has got a hydrophilic, a polar tail. It is a polar one, and this is non-polar. This is phobic. This is phobic, this one, and this is philic. This is philic. So it is amphipathic molecule, and therefore fatty acids form a monolayer. Now, we have in the entire epithelium villi are there. How come they go to the villi? They are not able to go there. Don't worry. What we have here, bile salt. Again come the role of bile salt. Bile salt again come for help. Bile salts. What the bile salts do? Bile salts convert them into small spherical balls. And these spherical balls, magically, they start moving in water. They are not floating. Bile salt convert them into spherical balls, and they start moving in water. So they can go to the villi and they can be absorbed through the villi. These small droplets, anyone who can tell me the name of these small spheres? Small spheres. Can you please tell me what these are called? They are not chylomicrons, sorry. They are not chylomicrons. If you want to see them, I'll try to show them. I'll show them. In my NCERT, I have annotated a picture. I'll show it to you. Digestion. Digestion and absorption. Yes. And I'm going to show it to you. Let us try the name. I'll just shout aloud the name of a person who write the name correctly. What these spherical balls all around hydrophilic. Hydrophobic is not, it has gone somewhere. It is a magic done by bile salt. Yes, please. Yes, here we come. And we don't come. ये क्या हुआ? Absorption, absorption, absorption. Okay. Disorders we don't have. Okay, no worries. Yes, please tell me the name. These are called Michels. Very good. These are called Michels. Perfect. These spherical balls are called Michels. Michels Obama. Michels. What has happened? Bile salts have converted into a spherical ball. All around the head is there. In the sphere, head is all around. And the hydrophobic tail is in the core. This structure is stabilized by bile salt. This is a Michel. And therefore, it can go to the villi and the mucosa, the villi, they absorb them by simple diffusion. So they can be absorbed by simple diffusion. They can be absorbed by simple diffusion, Question. Remember that fats, lipids are lipids. Membranes are also made up of lipids. So they don't charge any ATP. They are passive. Okay, they dissolve like simple diffusion. But please be careful. This is not the complete absorption. This is absorption only inside. Absorption only inside the epithelial cell. Inside the epithelial cell. This is only inside the epithelial cell. So something is confusing now. That okay, what is not confusing? This thing. Okay, it is forming a monolayer. What is not confusing? Bile salts convert them into Michels. Then Michels are everywhere in the small intestine and they are being absorbed into the villi. And the last part, when I said that it is only in the epithelial cell, this might be little confusing. But the confusion will be over after seeing this picture. I am showing one epithelial cell and I'll show you what happens to the bile salt, to the fats. Okay, now I'll show you what are these. These are the lacteals, lymph capillaries. A lot of gaps are there in the lacteals. These are the cells of the lacteal. This is a lacteal, a lymph capillary, and epithelial cell. Here is the lumen. It is the lumen, and in the lumen we have got fatty acid and glycerol, fatty acid and glycerol, and in the form of Michels, and they are being absorbed by passive diffusion inside. They are being absorbed by passive diffusion or simple diffusion. Now they are reformed again. They are reformed into triglycerides. They are initially they are digested from triglyceride only by lipase. Inside the cell, again they will form triglycerides. So 3 fatty acid plus 1 glycerol will become triglyceride inside the cell. This is a smooth endoplasmic reticulum where again they are reformed into triglyceride. Okay, why? It's all a strategy. It is a good planning done by the body. It's a strategy. Now this triglyceride will go to another organelle inside the epithelial cell. Identify which organelle is this. I have shown one cistern of this organelle. This organelle is smooth endoplasmic reticulum. You have to identify which organelle I am showing. Please tell me. Yes, these are Golgi complex. triglycerides come to the Golgi. Now the Golgi will, coat them with a hydrophilic protein. Golgi will coat them in hydrophilic protein. And now the work is done. Now these vesicles, the vesicle will be formed from the Golgi and they are ready for getting, this is exocytosis. You can see exocytosis, and through them the triglycerides, the triglycerides coated by hydrophilic proteins are out. You can see the triglycerides coated by hydrophilic protein are out. And now they can be easily taken into the gaps of the lacteals. And through the lacteal, now they become part of the circulation. First to the lymphatic circulation, and you know that all the lymph of the body enter into the veins, so they become part of the blood circulation. But so please understand, triglycerides cannot be taken into the blood or lymph directly, because again, this is non-polar and this is polar medium. Therefore, a strategy done by the Golgi complex, coating by a hydrophilic protein, and then it can enter easily. This is the absorption of fats, okay? And can you please tell me what these tiny droplets, these are protein coated, very tiny, tiny fat droplets coated with protein. What are these? Coated with proteins. Come on. What are these? Tiny fat droplets coated with proteins. Very good. They are chylomicrons. Very good. Everyone learn the name. These are called chylomicrons. These are called chylomicrons. Microns. What are these? These are small fat droplets coated by hydrophilic protein. These are small fat droplets coated by hydrophilic protein. Now you must be asking, sir, they were triglycerides, we digest them, again we reform them so that they can be absorbed. Otherwise, you cannot absorb them inside. You have to do this strategy. You first dismantle them, take them inside, again assemble into triglyceride, because again the problem here is it cannot be added into any aqueous medium. So Golgi complex, they will go to this end, which is called the cis end, and from the trans end, the vesicle come out. These vesicle contains tiny fat droplets coated with protein, and they are known as chylomicrons. And this is how the mechanism of digestion, a lacteal, and ultimately lacteal go to the veins. All the lymph of the body enter into the veins. This is the mechanism of digestion, of absorption of fats. Mechanism of absorption is IPND. This absorbed nutrients are taken up by the blood, by the cell, and they are utilized for the various activities of the cell. This is called assimilation. Still, lot of things remain undigested. We have not digested it 100% or completely. We don't digest fibers also. All the fibers remain undigested. This we have to remove from the body. This removal of undigested residue, this is known as digestion. And this is the last step of holozoic nutrition. We are just following holozoic nutrition: ingestion, digestion, then absorption, assimilation, and then digestion. The process of digestion is called defecation. The process of digestion is called defecation. Fecal matter, D, out, removing the fecal matter, defecation. Digestion is the process, digestion is the phenomenon, and defecation is the process. Now, defecation in human beings is an active process. Recall in the morning what you have done, what all you have done. You have removed your fecal matter voluntarily. It didn't happen that in the pants, it just came out. No, it happens in the infants, very little babies. But otherwise, then we have a control and we can do it voluntarily. But is it completely voluntary? Can we really control our peristalsis? No. That means it is a little complicated thing. We can control it voluntarily, because go to the anal area, anal sphincters. There are two anal sphincters, internal and external. Internal is involuntary, external is voluntary. It is a skeletal muscle, and we can control that. Oh, there is a maneuver by which we can control the way we are controlling our urination. The same maneuver can help us to control our defecation. So only when the right time, right place, and right posture we can defecate. It's a voluntary process. But as I told you, that it also includes a neural reflex. So defecation, I'm explaining you defecation. Defecation is the process by which egestion takes place. The undigested food is now collected into the rectum. It is, you know that the water has been absorbed and all the undigested, now it has converted into a semi-solid mass, coherent. Coherent means all has adhered to each other. It is just adhered, coherent mass. And this is actually increasing, okay? So as the day passes, this keeps on increasing, this mass is increasing, okay? Then what happens? Now it start exerting pressure on the wall, it exerts pressure on the wall of the rectum. Rectum wall has got stretch receptors. It is sensitive to stretch. The stretch receptors in the rectum are stimulated and they carry, so I'll write stretch receptors, they carry the signal to the central nervous system, they carry the signal to the CNS, to the central nervous system, and the central nervous system send efferent responses to the muscles of the rectum and also to the internal anal sphincter. So it wants the muscle to contract so that it can contract and the internal anal sphincter relax. These are the efferent signals coming from the CNS. This is a neural reflex. It's a reflex. It's a reflex action. It's a reflex action, reflex arc. Stretch receptors, they are stimulated, take the signal to the CNS, and the CNS respond by contracting. Now, whenever this happens, due to the reflex, what happens? No, defecation doesn't happen, but we get an urge. We feel pressure, we get an urge. And after this, we can go and we can voluntarily defecate. We can voluntarily defecate. We can voluntarily defecate. This is only to create an urge. This reflex is only to create an urge. It is only creating an urge. Hope you understand. It's a neural reflex. Receptors, afferent, CNS, efferent. There is an internal anal sphincter and an external anal sphincter. This is voluntary, it is involuntary. Involuntary is opened by reflex. Bachcho, I hope you understand. It is a reflex action. The reflex creates an urge, but removal of feces is a voluntary process and it requires mass peristaltic action. It requires mass peristalsis. It requires mass peristalsis. Mass peristalsis means large number of peristaltic waves. A wave which comes in such a way that it ultimately remove it out. It is not no. A mass peristalsis so that ultimately it will be removed. Otherwise, normal peristalsis keep on happening in whenever you open a live body in surgery, you'll find that the intestines will show some movements every time. During the anesthesia also, the intestine are showing some movements. But mass peristalsis, such a large number of peristaltic waves which ultimately remove it out, this is required for. So in defecation, few questions are there. Question number 1, what is defecation? Removal of the undigested weight out of the anus is called defecation. How it happens? It is a voluntary process. How the urge is created due to a neural reflex. And how it happens, it requires mass peristaltic movement. This is all about defecation. Very important, one or two questions you can expect, at least one question on defecation. And the question is based on exact lines of the textbook. But so I think this is easy, okay? Defecation, removal of fecal matter. And now I'll take you to the disorders. Okay, so disorders in my own way, it's very, very easy. Disorders so that you don't require to learn them again. Up to your NEET examination, you don't require to see them again. Disorder number one, question number one is, disorder number one is jaundice. What is jaundice? Yellowish discoloration of skin, of the sclera, of the body fluids. Why it happens? Doctors say that there is increase in the blood bilirubin, but it is a waste product. Bilirubin is a waste. Tell me the name of the organ which help in the excretion of bilirubin. Name of the organ. Yes, Suhas, your answer is correct. Windows 7, your answer is also correct. Name the organ that help. Very good, Karthik, your answer is yes. Very good. Boys, they pick it, student pick it up. And yes, the correct answer is liver. So liver help in excretion. If liver has some defect, excretion will be affected. And then what will happen? It will start increasing in the body, in the blood. And that is jaundice. So jaundice is an indication that there is some trouble in the liver. Okay? Jaundice. The second disorder is, yes, please, what was that? Vomiting. Vomiting means the stomach content come out through the mouth. Now, why this happens? This happens, there is a reflex called vomiting reflex. There is a vomiting center. We have in the brain a vomit center. In which part of the brain you have got a vomiting center? Come on. Which part of the brain you have got vomiting center? Yes, you are right. It is in medulla oblongata. It is in medulla oblongata of the hind brain. Bachon, whenever this vomiting center is stimulated, the person will feel, I'll write VC for vomiting center, and whenever this is stimulated, there is reverse peristalsis. There is reverse peristalsis, reverse peristalsis. As a result of that, vomiting occurs. So for vomiting, what happens? Reverse peristalsis. And for defecation, which peristalsis? For defecation, mass peristalsis. For vomiting, reverse peristalsis. Just remember that the vomit center is in the brain, and whenever any factor which stimulates that center, the stomach content come out of the mouth, and that is vomiting. This much we are supposed to know in vomiting. The third disorder I'm discussing here is diarrhea. Diarrhea means increased liquidity. You have heard about the liquidity of cash, but here is a liquidity of feces. The feces liquidity is high. Can you please tell me why the feces is more liquid than normally they have to be? Why the feces are more liquid? Where is the problem lying? Let us say this is the colon, rectum. Tell me, where is the problem? Why it is more liquid? The water, yes, you are right. Absorption is not proper. The water absorption is not adequate. So water is not absorbed, okay? And therefore, this is more liquid. And there will be abnormal bowel movements. So abnormal frequency of bowel movements. Abnormal frequency of bowel movements. What will happen? Again and again, mass peristalsis is occurring, and this liquid thing is coming out. And because it is liquid, it is very difficult for us to control it. We can control by external sphincter. If it is somewhat solid, it is liquid, it is very difficult to control. You understand the point? Abnormal frequency, liquidity, all is happening. Most of the reasons, most of the cases, there is colitis, inflammation in the colon, which is causing the diarrhea to take place. So very easy, diarrhea, okay. Absorption is at stake, absorption is a problem, okay, bachcho. Then in vomiting also, you can say that absorption is affected. Absorption of the food is affected. Absorption is affected. In vomiting also, absorption is affected. Bachcho, now the third. Next one, the next one is constipation. Constipation. So easy to understand. This is inadequate defecation. Inadequate. Inadequate defecation. Inadequate defecation. Why? There is abnormal bowel movements. Abnormal bowel movements. Defecation doesn't take place properly. Abnormal bowel movements. See, most of the time, what is happening? The problem lies in the bulk. Please understand, if the feces will not have bulk, the defecation is very, very imperfect. So to get the defecation easy, increase the bulk of the food. The food means the feces. You have to increase the bulk. If the bulk is more, the defecation will be very, very easy. There will be a lot of, a reflex, because this will exert pressure and then stretch receptors are activated and then neural reflex will be generated. But because the bulk is not adequate, therefore the neural reflex is also not at work. We try to defecate by exerting pressure by a maneuver, but that is also not sufficient. Okay, so increase the bulk. How we can increase the bulk? By eating more food? No, by eating the type of food which we are not able to digest, like fibers. If you eat more fibers, fibers are not digested, they will add up in the bulk. More the bulk, easy defecation. Okay, understand? So most of the cases it is due to lack of bulk, constipation, and bachcho, finally, a very important thing I'm going to discuss here, and that is indigestion. It's a very, very common thing also. Indigestion. What are the reasons for indigestion? I'm writing the reasons for indigestion, and this is what is important to note here. I'll write 1, 2, 3, 4, and 5, the causes of indigestion. Indigestion means inadequate digestion. It means inadequate digestion. What will happen due to that? What the person will complain? There is fullness of his stomach, as if he has just now taken food. His stomach is full, fullness he will feel. He will complain, tight ho raha hai, bahut sara ho raha hai, like, fullness. Why? Digestion is inadequate. So what are the reasons for this indigestion? Reason number 1, enzymes are not proper. So enzymes are deficient. Digestive enzymes are deficient. This can happen due to many diseases. This can happen due to some drugs. This can happen in old age. Enzymes are not adequate. Enzymes are not enough for digestion. So that is one of the reasons for indigestion. Reason number 2, overeating. Overeating. Now why this is happening? See, we have got the quantity, fixed quantity of enzymes now. God has already decided, programmed the body to make this much enzyme, for fats, this much enzyme. But if the food is more, enzymes are that much only. So extra food, problem of digestion. So therefore there will be indigestion if a person eats. If a person eats spicy food also, very spicy food, then also the indigestion, because many spices can inhibit the digestion process. Okay. The fourth and the most important reason, I will change the color to write that. What is the most important reason? Anxiety. Anxiety. Fight or flight. Fight or flight. Fight or flight. Triple F, okay. Sympathetic stimulation. Sympathetic stimulation. Sympathetic response. See, whenever there is a sympathetic response, a person is in anxiety or a fear or a fight or flight. right? Sympathetic will become active. Sympathetic nervous system, which is a type of autonomic nervous system, it become active. And remember, sympathetic inhibits digestion. Sympathetic inhibits digestion. Mali, sympathetic inhibits digestion. Once again with me, sympathetic inhibits digestion. Sympathetic inhibits digestion. Okay. Parasympathetic activates digestion. Parasympathetic activates digestion. The complete digestive system become active in parasympathetic. In sympathetic, the complete system, glands, sphincters, all are, inhibited. Anxiety. Please understand sympathetic response. This is a very common cause of indigestion. And finally, certain toxins can inhibit the enzyme secretion. Certain toxins coming out of the bacteria, for example, in food poisoning, in food poisoning, in food poisoning also digestion will not be proper. So enzymes are deficient, overeating, eating spicy food, anxiety, fight or flight, sympathetic response, and food poisoning. These are the important causes of indigestion. Remember, sympathetic inhibit digestion, parasympathetic, therefore sympathetic is called fight or flight. Parasympathetic is called rest and digest. Sympathetic, come on, sympathetic, fight or flight. Parasympathetic, rest and digest. Parasympathetic, rest and digest. So parasympathetic activates. But so these are the disorders. And lastly, I'll take some 5 minutes, properly 5 minutes, but I will make this concept such a clear in your mind, because whenever you work as a doctor also, you go to any WHO mission, you come across this problem. You know, what is this problem? Let us understand. A doctor is being appointed by a WHO, and the doctor has gone to certain places. A WHO, he's a part of a team, and the team is visiting certain areas. What are these areas? The areas are Southeast Asia, like Sri Lanka, the Southeast Asia, or Western and Central Africa. After that, they go to America and South America. In South America, these doctors find that the children suffer from protein energy malnutrition. Protein energy malnutrition, we call this as PEM, protein energy malnutrition. Please note down, PEM, protein energy malnutrition. The names are also taken from African language. The names, the two type of protein calorie malnutrition, we have got marasmus, and kwashiorkor. Kwashiorkor. My spelling is kwashiorkor. And CRT, I think, has written kwashiorkor. I don't know. Kwashiorkor, my mind is saying kwashiorkor. Marasmus. So two type. Now, what you have to actually learn, okay, this is not a general knowledge test, so they may not ask this thing, but this will be asked, that how they are different. But please remember, marasmus is less than 1 year of age, and this is more than 1 year. Okay, generally 1 to 4 years of age. Generally, babies, children, 1 to 4 years of age, more than 1 year. The main difference. In marasmus, there is extreme thinning, emaciation. Emaciation. We have a slang word in Hindi, a North Indian, not a Hindi word, but a slang in a North Indian language. It's a margilla. Very, very thin, emaciated. Emaciation means the same, extreme thinning. Emaciation means extreme thinning. So you will find this word in your textbook and CRT. It means extreme thinning, emaciation. Okay, what else you will find? you will find complete protein and calories, both proteins and calories, all mottam, complete malnutrition. And these problems occur whenever there is droughts, famines, wars. You just recall, now how you can recall? You can only see in the videos and movies what happens in the Second World War. We don't have much information about the First World War, but in the Second World War, in many countries, people died. People were sleeping many days without food, without proper water, without clean water, and they die of malnutrition, which is, in Western world, there is also many people are suffering from malnutrition, that is overnutrition. Obesity is obesity is the result of overnutrition. These are undernutrition, not eating food, no, and these are like when mother stops feeding in less than 1 year of age, mother stops feeding the infants. Infants means 0 to 1 year. Infants means 0 to 1 year of age. When infants, mother stop feeding. Why the mother can stop feeding? Number one, the mother is ill. Number two, mother dies. Number three, which is common, the mother has another pregnancy. These are the places where there is illiteracy, not proper information, not proper literacy, and the quality of life is very, very poor. The socio-economic condition is extremely poor, and therefore they don't have any idea about the contraceptives, and therefore usually the mother can also die, because if she is giving birth to 7th baby, 8th baby, 10th baby, so she becomes very, very ill, or else the mother had another pregnancy, and if a mother has second pregnancy, which is early, the breastfeeding stops. She will not feed now because the milk will not be formed. Okay, so these are the reasons the mother stops feeding the infants. So too early, when the milk, because 0 to 1 year, milk is the only food. You cannot replace milk. In marasmus, not only Margilla, you will find that they will have skin. They have thin and dry skin, thin and dry, and very wrinkled skin, thin, dry, wrinkled skin. You will find skin is wrinkled. The body looks wrinkled. You can count the ribs. There is no need of buying a skeleton. We can teach every bone. This is clavicle. This is scapula. This is this. We can teach every bone. They are so, you can just understand. We may have laughed on this, but actually you can understand the human beings born and sleeping without food. Even today, there are many places where the people are sleeping without food. In many Western countries, like I know about America, they have enough of crop to such an extent that every year they are dumping excess of crop in water. In the oceans, they have excess of rice and excess of maize. The crop is too much for them, and they can store, they can feed their people, they can sell wherever they can sell. But now the transportation cost is more than the cost of the crop. They dump the excess in water. Certain agencies like UNICEF and all, they are carrying them, or they are asking these countries to give donations, and they are bringing certain type of food to the people. But 0 to 1 year, this age is crucial, because even any food is not a proper food for the body unless the mother. Now, this is the kosher core. Just imagine a situation in a small town in an African country, where the UNICEF has sent lot of rice. It is a very poor quality of rice, but still food is there. They have sent rice in bulk. No other food is there, only rice is there. Some salt they have. What they are doing, they are boiling the rice, adding some salt, and eating morning, afternoon, evening, every day. The little babies who cannot feed, they take rice water, and they dip any cloth into it and they drop it in their mouth. Hungry children are licking that. So this is the situation. How do I know? I have read about them. I've seen in the movies, in the documentaries, docu-series, I've seen that. Now, when the rice is fed and they are hungry, so rice is, now they take rice because that is in excess. Free of cost rice is available. They are taking rice, only rice, only rice. They have enough of calories. Calories are in excess, but proteins are deficient. Then what will happen? A different situation altogether. Now they have got improper muscles, okay? So there is a wasting of muscles, but they have got excess of calories, and what they can have some fats are deposited. Certain fats can be seen. Some fat can be deposited, okay? These people have certain fat deposition. These people have extensive edema, vapu, extensive edema. Why? Because blood has less protein, and due to deficiency of protein, the osmolarity of the blood is less, and therefore the blood is losing water into the tissues, and the water is accumulating in the tissues, and it will have extensive edema. The belly, these babies will have pot belly. These babies will have pot belly. Okay, so kwashiorkor. These people will take only excess of calories but no proteins. Okay, and they will have thin limbs. Why the limbs are thin? Because of wasting of muscle. Wasting means degeneration. Degeneration means wasting. Wasting means degeneration of muscles. Degeneration of muscles, they have got thinning. The situation of the baby is somewhat like this. The face will be somewhat edematous. Okay? The face will be edematous. The tummy is a pot belly, and the limbs are very, very thin, very thin limbs. They have pot belly and they have got very thin limbs. Thin limbs are there. Okay? This is not fats, this is water deposited. This is the situation in kwashiorkor, and there will be growth of the growth rate is very, very low. Mental retardation. So mental and physical retardation occurs. Mental and physical retardation occurs. Here, skin changes can be seen here. This baby will be having a boat belly. This is having pot belly. This will have boat belly. Boat, just inside you can even touch the the vertebral column. Boat belly. Kwashiorkor, pot belly. Okay. Some fats in kwashiorkor. More than 1 year, kwashiorkor. Only protein, no calories, kwashiorkor. Full protein energy, marasmus. 0 to 1 year, marasmus. Extreme thinning, marasmus. Okay, here also thinning, but some part is edematous. Here it is extreme thinning, total. The skin is wrinkled skin, marasmus. Bachcho, this is all about protein energy malnutrition. I started with the geographical places like I told you, Southeast. Now, political turmoil is also one of the, turmoil means disturbance. Like there is a word called political turmoil. Turmoil means disturbance. To think about what is political turmoil means, you just see what is happening in Sri Lanka. That is political turmoil, and you know what is happening. They have a lot of malnutrition. They have a lot of problems. The economy is very, very almost negative. Okay, bachcho. So this is all. And calorific food values, as I told you, calorific food values. So you have to learn two types of food values. One is calorific. calorific values, calorific food values, and one is physiological food value. Physiological food value, calorific food value, physiological food value. Last 2 minutes. Calorific value, when you burn 1 gram in a bomb calorimeter. Where? In a bomb calorimeter. You obtain energy in case of carbs, in case of proteins, and in case of fats. So 1 gram in carbs, 4.1 kilocalories. Proteins, how much? 5.65 kilocalories. Fats, how much? 9.45 kilocalories. What do you have to do? You have to batti go to them. You have to mug up 4.1, 5.65, 9.45. There is no other logic here. You have to learn them. For only logic is carbs, they yield less energy than fats, but still, carbs are the most readily available source of energy. And the reason in biochemistry, or you can ask me any other day. Right now, I'll just complete the physiological value. Physiological value means 1 gram actually in the body. How much energy it is giving, okay? 1 gram in the body. So let me, carbs, proteins, and fats. Just learn 449. Very easy. No decimals. 449 kilocalories, and kilocalories. In carbs, 4. In proteins, these are physiological food value, 4, and fats, 9. Because the body is not that efficient. There are a lot of wastage can happen. In a bomb calorimeter, there is a complete combustion takes place. There is a complete combustion takes place in a bomb calorimeter. This is calorific food value, and these are, be careful, sometimes teacher play a game. Teacher say, how much energy is obtained in the body by 5 gram of fats? Tell me, 5 gram of fat in my body, how much energy we are expecting? Who will tell me the answer? Come on. 45 kilocalories. Good. Better with this, we come to the end. All the very best. God bless you all. And take it. And yes, your promise to me, your promise to yourself, anything which is mentioned in the NCERT, you cannot make any mistake in that. If it is in NCERT, you can answer it. Okay? I'm even not saying whatever I have taught extra. No, even I'm saying that. But whatever in the NCERT, that is your main promise. All the very best, children. God bless you. Class is over. For people online, they want, I can share the QR code. So much of preparation. ऐसा क्या कर दिया भाई? Yeah, got it. All the best, children. Bye-bye. Take care. God bless you. Bye-bye. Bye-bye.